Hey there, everybody. Welcome back to part 2 of chapter 17, our lecture on hypersensitivities, autoimmune and immunodeficiency disorders. So in part 2 here, we're going to talk about autoimmune diseases and some mechanisms of tolerance. So let's go ahead and just dive right in here. And in autoimmune disease, we have two types of immune disorders, right? We have autoimmune diseases where our immune system reacts against ourselves. So it's again, auto being self. So our immune system is reacting against us. Then we have immunodeficiency disorders in which the immune system is not functioning properly, or part of it is diseased or infected or not, maybe mutated. And so in an autoimmune, immune system attacks self. In immunodeficiencies, we have some kind of deficiency in the immune reaction. So autoimmune diseases, immune system loses what was referred to as self-tolerance. And self-tolerance is when we have what are called autoantigens. Autoantigens are self-antigens, things in our own body that our immune system will recognize as bad, say a mutated cell, and destroy it so that that cell does not enhance the mutation or promote the mutation of other cells. Because we start losing self-tolerance and we start reacting to these autoantigens, we can get production of autoantibodies. So we have antibodies, which are antibodies against things that we want to get rid of. And then we have autoantibodies. And autoantibodies are antibodies against our own cells. In what we refer to as self-tolerance, our immune system has built into it some self-tolerance. It has to recognize and understand what is self. So in self-tolerance, the immune system is like, oh, hey, that belongs there. I'm not going to react against that. So there are two primary mechanisms of what's referred to as self-tolerance. We have central tolerance. In central tolerance, we have what's called negative selection of lymphocytes that are going into the thymus. So in central tolerance, we're talking about T cells. And if a T cell recognizes an autoantigen, it's going to induce apoptosis, virus-infected cell, that sort of thing. Peripheral tolerance is where naive lymphocytes will actually see the antigen, but they don't receive any kind of co-stimulatory molecules during activation, such as B cells don't receive co-stims from T cells, or more than likely T cells do not receive co-stimulatory molecules from the antigen-presenting cells. In this case, the T cells or B cells will become what's called anergic. So in order for a T cell to become anergic, it is when it becomes a cytotoxic T cell becomes anergic, it will become a T helper cell, or it can become a T regulatory cell, and these cells will prevent self-responses. And this sounds a little confusing. But in central tolerance, we're looking at T cells not being primed properly. In peripheral tolerance, we're looking at naive lymphocytes being presented an antigen, but they're not able to respond against it. So they don't really get activated. So here on the right, we have just kind of a diagram that might help to kind of clarify some of what I'm trying to explain here. Central tolerance, bone marrow and thymus. This is occurring in the bone marrow thymus. We don't have, the cells are being primed, and they're going to they're going to react against autoantigens. In peripheral tolerance, our B cells are now in the lymph node. So this is occurring in the lymph node and in lymphatic tissue. In this case, cells are being they're being presented antigen, but they're incapable of responding. So they either become anergic, which is kind of a activated but non-effector cell, or they can mature and either go into apoptosis or go into T regulatory. So what are some of the autoimmune diseases that we're going to look at? Well, we have organ-specific ones. In organ-specific autoimmune diseases, we're going to look at type 1 diabetes, Graves' disease, and rheumatoid arthritis. In type 1 diabetes, there's some kind of destruction. In this case, an autoimmune disease will cause destruction of the islet of Langerhans cells, or what we call the beta cells, in the pancreas. And so cytotoxic T cells attack the islet of Langerhans, and this is going to destroy those insulin secreting cells. Once they're gone, they're gone. So they get destroyed, and then the person will then have to take insulin. So we get destruction of the pancreatic beta cells by cytotoxic T cells, and that's what's going on here in diabetes type 1. In Graves' disease, we have something affecting the thyroid gland. So you can imagine T cells are going to be involved in this, and in the thyroid gland, we end up with autoantibodies against what they think is an antigen on the thyroid, but what they're really attaching to is receptors that stimulate the thyroid stimulating hormone. This leads to hyperthyroidism, and ultimately, if left untreated, can actually develop into goiter. There's a pretty gross picture of goiter in your book where the thyroid just, it's a stimulating hormone, so the thyroid just grows out of control and they get that giant lump in the bottom of the neck. Hyperthyroidism is highly characterized by bulging eyeballs. This happens because of tissue and fatty deposits behind the eyes that start to push the eyeballs forward. Rheumatoid arthritis is a cell-mediated response, and autoantibodies are created against receptors and autoantigens found in joint tissue and sometimes in circulatory. It'll target joint tissue, and it is very similar to a type 3 hypersensitivity reaction, but these immune complexes continue to build up in the actual joints themselves, and so you can see here we get joint deformities. The thumbs are even a little off, and it can get really, really severe. The older someone gets, the more severe and kind of gnarled. The hands and feet in particular, but it can be seen in elbows and knees. And can be quite debilitating, and it's extremely painful. Now, there are autoimmune diseases that are systemic. We have organ-specific and systemic. In the systemic-specific diseases, we're going to look at systemic lupus erythematosus and myasthenia gravis. SLE, or lupus, is where autoantibodies are created against parts of our cell nuclei. So cells are lysed open, or as cells break down, we start to see antibodies attacking the nuclei, and we can get these kind of large complexes. Causes organ damage, rashes, joint pain, and swelling, and it is treated with heavy immunosuppressants. This rash right here is referred to as a butterfly rash, and because it has like this wing type of site to it. And so this butterfly rash is a key characteristic, a key sign of lupus. Myasthenia gravis is where antibodies bind to acetylcholine receptors. It causes paralysis. It disrupts the muscle reaction. So you can see here's our nerve cell. They're showing you here is where it would connect to muscle, right? Those acetylcholine receptors. But the problem we have is we have these antibodies get in here and bind to the acetylcholine receptors. Because those antibodies bind to those receptors, the muscle becomes weak, and there's some limited paralysis to it. Key sign of MG is drooping eyelids. The muscle is unable to hold the eyelid up, so we get this kind of drooping eyelid. It's treated with cholinesterase. Cholinesterase inhibitors. Cholinesterase is what enhances that acetylcholine uptake. So with cholinesterase inhibitors, what is done here is they will allow, instead of acetylcholine actually being taken into the cell, they'll inhibit the taking in, or slow down the taking in of it, to build up the amount of acetylcholine so that the acetylcholine molecules will outnumber the antibodies, and there'll be some type of signal. It won't completely get rid of the MG, but it will help to kind of diminish some of the effects of it. One of the problems that happens here is, just like we saw in the other autoimmune diseases, or in some of the hypersensitivities, is when we start having antibodies bind to something like these acetylcholine receptors, which is what they're binding to, we start activating complement. Sorry, there's a typo there. We start activating complement. And once complement gets activated, because of the inflammatory signals and the MAC formation, we can get cell destruction as well as get severe inflammation. So it can become somewhat painful, and ultimately the actual acetylcholine receptors will be destroyed. how do we treat autoimmune diseases? Well, the number one way to treat it is to kind of diminish the immune reaction to begin with. So we want to suppress the immune reaction. But the problem with immunosuppressants, which there are lots of immunosuppressants on the market, but part of the problem with immunosuppressants is it can make someone more susceptible to disease or infection. Examples of suppressing the immune system are immunosuppressants that target actual B cells or natural killer cells. They may target cytotoxic T cells. They can be cell specific. Most of the time we're looking at some type of anti-inflammatory medication as well. What are some alternatives though? Some alternative therapies include inducing tolerance. So perhaps trying to develop some kind of tolerance similar to what is done with type 1 hypersensitivities. Trying to reduce the immune system's reaction by introducing the antigen through a different route. Now, an example of this would be if somebody has like a rheumatoid arthritis or say they have MG, perhaps we can reintroduce the acetylcholine receptor or that particular epitope or autoantigen, introduce it orally. A lot of times this is done orally. One of the reasons that works is because of malt tissue. So remember, malt, we talked about earlier, about 2 weeks ago. Malt is that mucosal associated lymphatic tissue, and it's this kind of new part or third party in that immune system. So if we can introduce it to malt, we might be able to get to institute or increase tolerance or create some tolerance by introducing the antigen through a different route. Now, there are a couple of different types of autoimmune diseases. The four basics are the complement dependent, where antibodies are attaching to cells, thereby activating complement, and complement creates a membrane attack complex and the cell lyses open. Then there is antibody-dependent cellular cytotoxicity. So the ADCC reaction is another mechanism by which autoimmune diseases destroy cells. Then we have antibody-dependent cellular phagocytosis. Remember, when antibodies bind, one of the effector functions of antibodies binding is opsonization. So we get increased opsonization and ultimately antigen presentation. And of course, apoptosis. If a particular type of antigen is attached, the cell is going to enter into apoptosis. Now, one of the things I want to talk about is this is actually a diagram for a drug called rituximab. I think I said that correctly, but I may have pronounced it wrong. So this particular immunosuppressant works on all four of these different mechanisms of autoimmune diseases. For example, over here in our in complement-dependent cytotoxicity, rituximab is a monoclonal antibody. Remember, any drug that ends in MAB is a monoclonal antibody. This antibody will attach to the Fc region of IgGs. So in this turn, rituximab will actually bind to the receptors that are activating complement, the antibodies, the autoantibodies that have activated complement, thereby, thereby preventing this cell lysis. Then we have the ADCC reaction. In this case, the antibody, this monoclonal antibody, binds to the antibodies that would activate the natural killer cell. So the natural killer cell you can see here, NKC, can't bind. Here, the cell-mediated cytotoxicity. Rituximab keeps it from activating complement. And the ADC phagocytosis, it's going to prevent macrophage phagocytosis, so the Fc receptor of macrophages will not be able to bind. And then, of course, apoptosis, it may block the receptors. It can bind to the receptors known as the CD20s that would receive signal from macrophages or other cells for apoptosis. So this is just one immunosuppressant, but you can see as a monoclonal antibody, by binding to Fc regions and such, we can actually prevent a lot of other immune reactions from occurring. But you can also see why this would be, as a immunosuppressant, why this might open someone up to further infection, because now we're limiting the immune system's response. So here are a few other autoimmune diseases and some of the treatments that are used for them. We're not going to go through all of these. You don't have to memorize all of these. I just wanted to let you to let you see that there are a couple of different types of autoimmune diseases or immunodeficiencies. There's a little bit of both here, and there are different treatments, stem cell treatments, gene therapy. Those are treatments that are definitely being researched for all of these. But if we look at antibody deficiencies, which so this would be an immunodeficiency, we can replace IgGs. We can give antibiotics and antifungals, and of course biological agents to suppress inflammation. Here in autoimmune and autoinflammatory disorders, immunosuppressants and corticosteroids are usually the treatments of choice. There are a few biological agents like the rituximab that I showed you, which are your monoclonal antibodies. So to review autoimmune disorders, make sure you know what an autoimmune disorder is, what are the two classes, describe one organ specific and maybe one systemic, what are the role of antibodies, what immune reactions do lupus and rheumatoid arthritis have in common, what are the three forms of treatment for these disorders, and what's the theory or concept behind immunotherapy for immune tolerance. How do we develop immune tolerance? What's the idea behind it? So now we're going to talk real quick about some immunodeficiency disorders. So here we have the opposite. Instead of our immune system overreacting like an autoimmune disorder, now we have our immune system not acting enough. What is suppressing my immune system? What is keeping it from reacting against both self and foreign antigens? So we have a diminished capacity to respond or a loss of systemic reactions. There are two different types of immunodeficiencies. There are primary immunodeficiencies, which are inherited, those that you are born with, and then there are secondary immunodeficiencies, which are those that are acquired later in life, usually through bacterial infection, viral infection, environmental exposure. There's a couple of different types of immunodeficiencies. When someone has an immunodeficiency, they are far more susceptible to infection, both bacterial, viral, fungal, and parasitic, than your normal healthy person. And this is, of course, because their immune system cannot respond completely. So here are a couple of different just examples of some origins of immunodeficiencies. They could, in a primary immunodeficiency, that could start, of course, it be in the bone marrow, and then this develops into blood cells that are not functioning properly. Here's de George's syndrome, which is an issue with the thymus. Thymus is not functioning properly, so T cells are not primed correctly. ADA efficiencies, recurring bacterial infections, all of these are effects of these different lymphoid immunodeficiencies. So in the primary immunodeficiencies, we have four basic classes. We have antibody immunodeficiencies, defects of phagocytic cells, lymphocyte deficiencies, and defects of complement. In antibody immunodeficiencies, IgA is most commonly the antibody that is missing or that is reduced. It usually results in severe mucous membrane infections, which is unfortunate because mucous membranes are the primary portal of entry for most pathogens. So without IgA antibodies neutralizing these pathogens before they can take hold, you'll see a lot of mucus type infections. So these would be gastrointestinal infections, respiratory infections, upper and lower respiratory and urogenital tract. One example, aside from IgA immunodeficiencies, is one called A-gamma globulinemia, and this is where a person does not have the ability to produce any antibodies, and this makes them severely susceptible to infection. With defects of phagocytic cells, there's one called chronic granulomatosis disease, where phagocytic cells lose the ability to phagocytize. They lose the ability of their FC receptors their FC receptors are mutated, so they don't recognize antibodies being bound and those sorts of things. They may not recognize complement. So if their receptors are not functioning properly, they may try to bind to something, but they can't phagocytize it. They don't take it in. This can cause the formation of granulomas within tissue and organs, and these are really large immune complexes that begin to disrupt organ function. Lymphocyte deficiencies are exactly that. Lymphocyte cells are not functioning, so those are, remember, your B cells, T cells, and natural killer cells. In severe combined immunodeficiency, SCID, a person has no lymphocytes, so no T cells, no B cells, no natural killer cells. They are born with this, and a bone marrow transplant will be required shortly after birth in order to make sure that they can produce some type of lymphocyte. De George's syndrome is when somebody is either born without a thymus or a thymus that is not non-functional. And so in this case, if there's no thymus, the person will have no T cell response. They will be very susceptible to like viral diseases and intracellular bacterial diseases, but yet their B cells are still working, so they may still have some antibody response to bacterial diseases. There's just not going to be a lot of memory cell production. So they don't get a lot of long-term immunity, but they do get some short-term immunity in certain cases. Then we have defects in complement. With defects in complement, we often see immune complex disorders. Complement is not following the proper cascade mechanism. It's not activating its next protein properly. There can be missing proteins, so there's a mutation in a gene for one of the complement proteins, so it's either missing or doesn't function at all. And this can lead to, depending on which complement protein it is, this can lead to a whole host of different signs and symptoms, and of course different infections. Somebody with defects in complement are going to be highly susceptible to, particularly to bacterial infections, simply because complement really is one of those first lines of defense. Remember, complement is part of the innate immune system. In secondary immunodeficiencies, these are immunodeficiencies that somebody develops after birth. So it's not something you're born with. There's no genetic mutation. It's something, it's either through infection or exposure to something that has suppressed the immune system. Now, there are 3 very common causes of weakening of the immune system. The first is drugs, whether they are pharmaceutical or legal or illegal, but medications and drugs do weaken the immune system. Next is infection, bacterial infection. If you have an illness, you have contracted a disease, you are going to be more susceptible to other diseases because your immune system is already stressed and taxed. So your immune system can only respond to so many things at once. So infection does temporarily usually weaken your immune system. If it's certain types of infections can sometimes lead to permanent immunodeficiencies. Just depends on the organism and the immune response in patient. Another really important cause of suppressed immune system is malnutrition. Malnutrition is a really big problem, not just here in the United States. It is a problem here in the United States, but it's an even bigger problem in several other parts of the world. And malnutrition can lead to poor development, lack of proper physiological function, and of course exposure to many pathogens as well. So malnutrition is a big problem. The healthier somebody is, the better they eat, the more effective their immune system is going to be. Cancer is a secondary immunodeficiency. That's the result of genetic mutations in our cells. And when somebody is actually diagnosed with cancer, it is considered a failure of the immune system. Your immune system should be, your cytotoxic cell responses should be detecting changes in cells and destroying them before they can develop into cancer or tumor. But when they get those cells develop too quickly or too rapidly, or the immune system doesn't recognize it for some reason or other, it can develop into a cancer or tumor. Cancers of the lymphatic system, things like leukemia and B-cell lymphomas, those sorts of things, are considered secondary immunodeficiency diseases. Then we have viral infections in the immune system. And of course, this is any virus that attacks any part of the immune system. Of course, the most infamous is going to be HIV, or human immunodeficiency virus. It is called an immunodeficiency virus. In cats, they call it feline immunodeficiency virus. And in monkeys or in primates, they call it simian immunodeficiency virus. Measles is another one. When somebody gets measles, they're going to be in an immunodeficient state because measles replicate inside of immune cells, similar to HIV. except the HIV is strictly to the CD4 T lymphocytes, where measles is like macrophages, dendritic cells, neutrophils. Syphilis and leprosy also are diseases that can lead to immunodeficiency if left untreated. So in reviewing immunodeficiencies, make sure you know the difference between an immunodeficiency and an autoimmune disease. The two types of immunodeficiencies, be able to maybe describe one or two examples of each that we talked about in the lecture, describe the 4 classes of primary immunodeficiencies, and then answer these questions here. Be able to describe the class or name the class of immunodeficiency a patient would have with specific signs or symptoms. 3 most common causes of acquired immunodeficiencies, and then people with B-cell infections more prone to bacterial infections, and those with T-cell deficiencies are more prone to viral infections. Why? Why is that? Look at the mechanism of those two and understand the function of the immune system and how when we suppress one part of it, how it affects other parts of the immune system. So that concludes part 2 of chapter 17. In part 3, we'll look at some immune disorder treatments. See you in class.